Hi everyone, my name is Gunnar, and I'm so happy to be here. I've been coming to this meeting for quite a while, and I'm very interested to share our work. Don't have any disclosures. So we just heard about how AI, and this is going to predict and change everything. I hope that's the case. Mouse modeling is difficult, but I think in the context of this disease, I still think having at least a single model that you can try things on might be useful. I'm kind of struck by the progress that's been made in Alzheimer's disease and Parkinson's disease. Not that those direct mouse models have come out with therapeutics, but the mechanisms, the work, the research that goes into that, they've moved the field really far. So I think there's a place for everyone. I'm also going to talk about sort of a serendipitous trial that we just we started based off of the collaboration with experts in the field, and we'll go from there. Avik Roy is the Chief Scientific Officer of Simmaron Research. He's based in Milwaukee, Wisconsin. That's his laboratory. He's been leading this work. My lab is at the Indiana University, and that's where I am. So we have two different PIs doing two different jobs. A division of labor is useful as the chief sections, as we've said. Avik is sort of leading on the scientific side. I'm leading more on the clinical science side, so working directly with doctors and doing my best to help get samples. So just a quick warning, all these patients are different in this field, but there appear to be some subgroups. This work here doesn't intend to hypothesize what could be the cause. Hopefully AI will figure that out. We're focused on addressing cardinal symptoms that we know exist: post-exertional malaise, dysautonomia, cognitive impairment. And I think observing a response to a therapeutic intervention in a living, breathing human being is really important. Okay, so we have been interested in autophagy. My previous world and Avik's previous world comes from Parkinson's disease and Alzheimer's disease, where autophagy has a critical role in the removal of depolarized mitochondria. It also, and metabolically inactive proteins. Over the past couple of years, or more than that, we've known that ME likely has some type of mitochondrial pathology. It's likely not a lethal type of mitochondrial dysfunction. If that were the case, we wouldn't be seeing this. It could be an issue with the inability to renew and create new mitochondria, and just simply house maintenance. You can think of autophagy quite simply as one of the primary recyclers of mitochondria in the area, in the cells. So what we first did to get going with this, I had worked for a long time with a clinician, group of clinicians, Dan Peterson, some of you all are here, others, and we decided to start screening, looking at autophagy, because we had not seen people really take a deep dive into that. And interestingly, we found a group. It's small, but it's a significant group. Again, subsets. This isn't everyone that had this serological evidence of autophagy disruption. We identified the marker being phospho-ATG13, which is one of about 49 or 50 proteins, if not more, that participate in the autophagy-coupled lysosomal biogenesis pathway. So we identified this in the serum of patients, indicating that it's in its phosphorylated status, just meaning that this was not where it's supposed to be. It should be inside the cell. We shouldn't be finding this in the blood, presumably. ATG13, autophagy-related gene 13, is one of the rate-limiting steps for the initiation phase of the early autophagosome. If you're aborting autophagy early, you're going to have a lot of problems with the removal of depolarized mitochondria. Since we found it in the serum, we also identified through a series of other experiments, that phosphorylated ATG13 can directly stimulate the expression of reactive oxygen species, nitric oxide synthase in human microglia cells. And we assumed that because this is small enough, and we've seen too that it can cross the blood-brain barrier, it can directly activate these microglia cells, the host immune cells of the brain, via the RAGE receptor, the receptor for N-glycation. So we were trying to figure out, well, what would drive this ATG13 inactivation? Because autophagy impairment can result in mitochondrial dysfunction, cell death, inflammation. And upstream of the ATG13 protein, the kinase, which phosphorylates, is known to be the mTOR, mammalian target of rapamycin. And so we had this idea and have continued to work on this out to identify chronic mTOR activation in a series of patients. And to do that, because we don't have an AI-based machine, we did some animal modeling, old school. And again, we're trying to model these, when you're doing this work, I've just gone on about it, I don't need to kill it over the head, but we have two different models, and they're for different reasons. One is to identify, what is going to happen if you actually feed an animal like this, an animal, mouse, mTOR, an mTOR agonist. The other is looking at what happens if you can conditionally knock that down in specific tissues. So we got these animals, and we're at the University of Wisconsin at Milwaukee, and we work with a bunch of chemists, so we have the ability to make compounds, and we developed a compound, MHY1485, which is a mTOR activator. Fed the mice this, just black mice, normal mice, for 14 days, and we wanted to see if we could identify any evidence of muscle fatigue or post-exertional malaise. Those seem to be the two that were really there. So these are the mice after 14 days. They don't look like normal mice. They, this type of sort of shivering, and we have a light over them. Normally they're trying to avoid hawks, so they'll go hide in the corner. We were surprised that, not totally unsurprised, that a compound like this would result in a pathology, but we were surprised at the level of it, essentially. One of the things when you're animal modeling, you don't want to kill the animal. So we did notice that while this was a pretty debilitating phenotype, like you just saw, there was, not a significant. There was no death, and there was only a little bit of reduction in weight for the females versus males. We did EMG recordings on the biceps to try to figure on the bicep foramus and the muscle tissue to try to figure out if we could identify abnormal EMG patterns, which we did. We saw a reduction in the grip latency in a dose-dependent manner. So when the mouse hangs from the cage over an area, it has a, it doesn't want to fall. And so, but the increase of this, in increasing doses of this mTOR agonist, these mice were unable to hang on so long. We also saw a difference in gross locomotor movement. To model the 2-day treadmill PEM study, which we know is one of the diagnostics for chronic fatigue syndrome patients and identifying PEM, we again borrowed an idea from what happens in humans by looking and running these animals on a treadmill. We saw a pretty prototypical response where you have on the second day right after running on the treadmill, you see a rest period, which is this middle bar. These mice are tired. This is right after they've been running. Now they're tired. But then we waited for 2 days, and we saw that the SIM1485 fed mice, displayed a pretty not respond. They didn't return to their normal state of exercise, whereas the vehicles did. We did a series of protein analysis in skeletal muscle to identify that, yes, we were touching mTOR, and yes, that mTOR was upregulating this phospho-ATG13 species. We went through further and looked at specific immune activation. We wanted to see if there was anything going on here, so we took and looked at the muscle tissue, and we did see some mononuclear cell, a significant amount, and we thought initially that might be T-cell mediated. We went further and identified it was actually based in the macrophages, specifically CD40 IBO1 positive macrophages, and that those macrophages, in addition, were releasing a pretty significant amount of reactive oxygen species, which is kind of what those things do. So simply by feeding these black mice this mTOR agonist, we start seeing some evidence of post-exertional malaise. We start seeing evidence of some of the pathological findings that have been observed in humans. We saw a similar, when we started looking at the neuromuscular junction to identify what's happening with the nerves, we did see a reduction in myelin basic protein, TUJ1. We saw dissociation of the myelin layer and also some tunnel bodies. So this kind of gave us some confidence that in addition to having some physical phenotype you could watch on the treadmill and you could see the animals at the level of the muscle simply upregulating and pushing mTOR resulted in this phenotype. Long story short, one of the way to really identify ATG13's involvement, which is the serological marker that we identified in the patients, you have to kind of knock that down at the genetic level. So we did just that. Right now, we've created a conditional knockdown of ATG13. So what that means is that the animals, ATG13, if you were to knock this piece out it would be lethal for the most part. And then if you even do it in a hemizygous form, that results in some other issues, like they develop cardiomyopathy and they're smaller. So to do that, we did a tissue-specific conditional knockdown where we pre-floxed the genes and specifically are knocking out ATG13 in tissues. It also allows you to have the mouse come to term, grow up completely normal, and then induce the global knockdown of, or the tissue-specific knockdown of ATG13. Here, we're seeing a similar pattern with this knockdown animal. So this is showing the same sort of PEM pathology and also just a general reduction in the total movement when you force these animals around the treadmill. Okay, so fine. Now we've sort of looked at and we want to try to modulate mTOR. Well, then we came up with rapamycin, kind of. But really, this has been a field that's kind of exploded for a long time. There's a specific, this has been looked at by many people in the context of anti-aging. And in fact, we decided to kind of reach out to some of the colleagues that we have and let's just get to it. And we found some people that were already doing this. So David Kaufman at the Center for Complex Diseases, he approached me last year and he said, hey, you're working on chronic mTOR activation. I've been giving my patients rapamycin for a couple of years and they're feeling better. And so we said, okay, well, let's try to get this group together and do a clinical trial. We have done that now. This is sort of the schematic for how patients are recruited, the type of work that they're going to be doing, the timelines. We have different collection time points, so we're collecting at the baseline phase and then throughout the treatment phase. One of the interesting things with this project and with rapamycin in general is sort of the ramped dose approach. A lot of these folks with ME/CFS, they have multiple chemical sensitivities, so being able to ramp the dose in a single milligram format over the course of the ramp period we're hoping, will continue to help keep people on the drug longer. After you go through this ramp phase, there's a period of continuous dose for 18 weeks followed by a 4-week holiday, and then you can either repeat the ramp or go back directly to the effective dose. And it's important when I say effective dose, we chose 6 milligrams as sort of the max area. Already in the patients that we've tried this with, we've seen that some individuals are responding much better to sort of a lower dose. Again, the long half-life of sirolimus, rapamycin allows for the weekly dosing. We're doing standard lab checks to make sure that people are doing okay, and we're also making sure we're taking a look at lipid panels as that can be affected through chronic sirolimus use. But I think it's an important idea whenever you're doing an intervention in ME/CFS that you can kind of go slow and go low and slow instead of just hammering in. And I think with this type of protocol, it makes it a bit more user-friendly for individual patients. So what we're doing, this is the short phase piece that we've just started. We're looking at 3 different time points. After the baseline period, we're taking study samples. These are listed here. Our primary endpoints, and this is really important, is the REDCap data. These are the self-reported questionnaires that we're hoping to see some type of response. And we're also looking at safety labs, obviously. For our work, we're interested in this autophagy screening array. We feel that if we can predict individuals who are going to potentially respond, one way to do that would be to test them and compare data before and after. We have a series of other stuff we'd like to look at with this biobank. Maybe we could give it to your company and they could take a look at it too. We've seen this already. We've seen people, this is not everyone, we want to be clear that everyone is going to be a little bit different, but we are seeing subsets. So it's encouraging to see, especially with bringing in new groups and new physicians, new cohorts, that there is still people, and this is a true thing. We're not crazy. What we're really interested in now is looking at the REDCap data to try to correlate some response over time. So we've got like 104 people going right now at 5 sites. We're going to finish with this short phase really quick, and then move on to continue to enroll people. So some of the pros of the rapamycin, I think whenever you're thinking about getting drugs for chronic fatigue syndrome, I think you want to look at something that is probably off patent, just to start there. I think new drug development in this field is going to be difficult because of the costs associated with that process. So there's a lot of great candidates out there that I think can be useful. I think it's also important to look at something that can be managed at the level of primary care, that's accessible. This is a chronic disease. It needs to be, hopefully, a cheap drug. That would be wonderful. Something you could blind and with, again, accessibility. The immunosuppression piece, I think that is out there in the literature. I think, even in the anti-aging world, they've taken a look at this. The dose and the frequency of the dose sort of mitigate that for the most part, but that is, again, something to consider when looking at this. The lipid panel changes are known to be there, but we do think that might be an indication for clinicians to back off treatment or reduce the dose. Just to be clear, this is not a controlled trial. David Sindstrom's going to talk about a controlled trial coming up. And only some of these people are going to respond. We're really just trying to take a first stab at this, and this is our best effort. So I think the other thing that's important is a decentralized clinical trial model. That's what this is. So we have everyone is staying at home. They're not coming into the clinic to take a look at, to be a part of this. They can participate from home. Kits get shipped out to them. FedEx is a wonderful company. I'm always so impressed by how they work, and it's great to have them on board. And we do have mobile phlebotomy. I think that's an opportunity for more people to participate, particularly the most severely affected and ill. We don't have an exclusionary criteria at this point because we're trying to see if we can predict who's going to respond through this ATG13 screening assay. So like I said, we're going to go through the short phase here and get that done, at least with enrollment, as quickly as possible. What's nice about this is we can then continue to follow these people for a longer period of time to see if this response, if there is one, is sustained over a period, and we'd like to continue to expand other clinics. And if we had an unlimited budget, I think there are some things we would do, but we don't, so that's sort of where we're going. I'd like to thank everyone so much for having me at this meeting, and all of the people that I work with. I'd really like to thank the patients. It's incredible just how dedicated they are to this project, and we will see this through, and we will publish whatever we find. Thank you very much.